All right, so I'm back on this model on page 78, and I'm going to be adding my bases to this DNA strand. So I am going to get started by looking at the little pieces that I have cut out. I need a big one that has a point. So this one right here is one of the large ones. If it's a large one, there are only two possibilities for what it can be. What are the two possibilities? Good. You could have said an A or a G. We're going to make this one an A, and I'm going to tape it in place. So make one of those circle tapes, and then we're going to tape it so that it's right off of that carbon number 1, and I'm going to label this one with an A. All right, next, let's grab another one. Let's pick another large one. If the first large one was an A, then this one must be a G, and I know that it's a G, not an A, because it has a different end. So these are not the actual shape of a nucleotide, but the end here tells me that it's not an A. A has a point. G has this rounded, circly thing, half circle. All right, and so I'm going to attach this one right here and label it with a G. So now I have a whole nucleotide. So let's go ahead and label one of these whole things as a nucleotide. So I'm going to start up here, and it's going to include that phosphate. It's going to include this G, and then it's not going to include the phosphate below. So what I've just drawn here, this box, is surrounding a nucleotide. So this is one nucleotide. If nucleotides are a phosphate attached to a sugar attached to a base, then up here I really should have a phosphate up here. And I can draw my phosphate in if I want to. You don't have to, but I would have an H on this side, and then my C would be bonded to an O, bonded to a P, and it would look like that. So I'd have my phosphate up here. Now this phosphate, this sugar, and this base makes up a nucleotide. And I could have this phosphate and this sugar and a base over here make up a nucleotide. But first, I'm actually going to try and make the other side of this DNA structure, because we said at the beginning that DNA is double-stranded. Well, if it's double-stranded, I only have one strand right here, and I have to have another strand over on this side. So I'm going to start by laying out my pieces. I've got my sugar, I've got my phosphate, and the base that's going to attach to this one is going to be this base here. And that one's going to be my T, because those are going to fit together. But this does not fit like this. The only way that I can actually get this to fit with it still having the same nucleotide structure is to actually turn this whole thing upside down, because the nucleotide has to touch, the nitrogenous base has to be attached to the first carbon, and the phosphate has to be attached at the top to this 5th carbon. So what I'm going to do is I'm actually going to turn my notebook around so that I can make this easier on myself, because I have a really hard time working upside down. And I'm going to start putting these things in place. So first I'm going to need to tape that nitrogenous base in place. So I made my tape too big. So now that I have my nitrogenous base in place, it's across from my A, so I've got my A. Now A binds with T. And how many hydrogen bonds are there between A and T? If you said 2, you are correct. So I'm going to draw my hydrogen bonds. Now, my sugar has to attach off of the first carbon. So I'm going to make another circle tape. I'm going to take my sugar here, and I'm going to make sure that carbon number 1 is attached right to the center. Oop, it's a little crooked. Of this nitrogenous base. Now I need to label this so that I can actually see that everything is connected properly. And my phosphate is connected at that 5th carbon. So now I'm going to add in my phosphate with little tiny circle tape. Okay, and I'm going to put my phosphate in here. I'm going to use a different color like I used before. So P, double bonded O, O minus, O, and then there's a bond right there. Alright, so now I've added my first nucleotide. I'm going to need to add my next nucleotide. Now G binds with, if you said C, you've been following along. And I'm going to need to make circle tape. And I'm going to attach the C across from the G. Now, DNA is a very particular structure. All the pieces are the exact same size and they're going to be the same distance apart. So I need to try and match this distance here with this distance here. And that's going to make it so that everything's actually going to fit and line up. If something was a different size or a different shape, then it actually wouldn't fit together. Things would be bent and at angles, and those bonds would be stretched and they would break, and it just wouldn't work very well. So let's label this one C. And how many bonds are there between C and G? There are 3 hydrogen bonds. So I've gone ahead and I've drawn my 3 hydrogen bonds. And now I'm going to add the sugar and the phosphate to finish out this nucleotide. Alright, so it should work where I can put my sugar and it should attach to the phosphate at the 3rd carbon, and it should attach to the nucleotide. And it wasn't, attached to the nitrogenous base. It's not totally perfect, but it's close enough. And then I'm going to add my phosphate up here. And I'm going to go ahead and label everything. And one of the reasons that we're doing this so many times, even though I could have just given you a printout, is because you actually need to be able to build this structure. So one of the things that we're going to be doing is we're going to be making models of DNA. And when you're making those models, it's really important that you make them correctly. And so we are putting this together now so that you can see how this works. Okay, I'm going to continue to build my backbone going up. So just like I did it here going down, I'm going to build it here going up. So I know that this phosphate is going to be connected to the third carbon. And then that there's a carbon here, which is going to connect to my next phosphate. And then I'm going to make another one. So sugar. And phosphate. Okay, now the sugar phosphate backbone here and the sugar phosphate backbone here are connected in the middle by those nucleotides. So, gosh, I keep saying nucleotide when I mean nitrogenous base. So, sorry about that. They are connected by nitrogenous bases. So let's go ahead and add the bases in the middle. Now, it does not matter which base that I have remaining that I put here, as long as I match it properly. So I am going to put my G right there. So I'm going to make my tape. Alright, so my G is going to go here. And if my G goes right here, then that means that I must have a C across from it. And if I made these all the right size, then that C should match up with the carbon. And I'm just going to draw a little line to show that these things are connected, because it didn't actually work out that great. So this is G, and this is C. And there are 3 hydrogen bonds between them. Okay, we've got one more. Again, it doesn't matter which thing I put on which side. So let's go ahead and I'll put the T on this side. And that means that I'm going to have my A on the other side. And it's this sequence of A's and T's and C's and G's. It's the order here that's really, really important, because it's going to be the order that makes it so that you have your genes and I have my genes. So the order of the bases is what makes those different proteins different proteins. Remember that DNA codes for proteins. So if this one is my T, that means that this one is A. Okay, there's one very last thing that we're going to go over with this. And the very last thing that we're going to go over is the directionality of these strands. So I'm going to turn my notebook back around. And we've got a direction that these strands are going in. When I say a direction, I mean one of them is pointing this way and the other one is pointing this way. And that's just the way DNA works. DNA is arranged so that one strand goes in one direction and the other strand goes in the other direction. And the way that we talk about direction with DNA is we talk about it in terms of what we call 5' and 3'. And the 5' end of my DNA is the end of my DNA that has the 5th carbon pointing up. So remember, I start at the oxygen. 1, 2, 3, 4, 5. That means this end is the 5' end of this strand. The 3' end is this end going down, 1, 2, 3. So the 3' carbon is here. And you'll notice that that means that the 3' carbon is on this end, and it's on this end, and it's on this end. So this is the 3' end of this strand, and this is the 5' end of the strand. Now on this one over here, I still start at the oxygen, and I still count clockwise. So 1, 2, 3. This is going to be the 3' end. 4, 5. This is going to be the 5' end. If I go to the top of this, I can do the same thing. Start at oxygen, 1, 2. This is the 3' end of this strand, and that means that this is the 5' end going this way. So what I would say here is that I have this strand is arranged from 5' to 3', but this strand is arranged from 3' to 5'. So I'm going to go back to my first model. If I label this strand right here and say that this is the 5' end, then as I go down to the bottom end, this must be the 3' end, because it always goes from 5 to 3. Now on the other strand, I know it's going to go in the other direction, which means this must be the 5' end, and if I follow it out, that means this end must be the 3' end. And that's how all of this is going to work. So I'm going to label one more. This one here. This end up here, this has to be the 5' end of this nucleotide and the 3' end of the nucleotide, because this is the 3 carbon and this one here is the 5 carbon. And that is DNA structure.